Okay, so formation of blood cells, the process is also known as hemopoiesis or hematopoiesis. So the primary location or primary site of hemopoiesis is the red bone marrow, but during early life stages, like when the growth embryo is growing inside the womb of a mother, in that time there is a change in the site of the blood cell formation. Okay, so earlier on it happens in the uh, yolk sac of an embryo, and later the site shifts to the liver, spleen, thymus, and the lymph nodes of the fetus. So the red bone marrow becomes the primary site of hemopoiesis in the last 3 months before birth, and it continues as the source of blood cells after birth and throughout the life. So, what is the red bone marrow? Red bone marrow is a highly vascularized connective tissue located in the microscopic spaces between trabeculae of spongy bone tissue. Okay, so it is present chiefly in bones of axial skeleton, pectoral and pelvic girdles, and the proximal epiphysis of the humerus and femur. About 0.05 to 0.1% of the red bone marrow cells are called as pluripotent stem cells. So the pluripotent stem cells which are shown over here, uh, they constitute around 0.05 to 0.1% of the red bone marrow cells. Okay, so these cells are also known as hemocytoblast, and they are derived from the mesenchyme. So we have studied about mesenchyme when we have studied the connective tissue. Okay, so this is mesenchyme is the tissue from which almost all connective tissue gets developed. These cells have the capacity to develop into many different types of cells. So you can see the type of cell which are primarily shown over here, that is the myeloid stem cell and the lymphoid stem cell. In case of newborns, all bone marrow is red and thus active in blood cell production. So, as the aging happens, the rate of the rate at which blood cell formation happens, it decreases, and the red bone marrow in medullary, that is marrow cavity of long bones, become inactive and is replaced by yellow bone marrow, which consists largely of fat cells. Okay, so as it is consist of the fat cells, that's why it appears to be yellow. That's why it is called as yellow bone marrow. Okay, so under certain conditions like severe bleeding, the yellow bone marrow, it can revert back to the red bone marrow, and this occurs as blood forming stem cells from red bone marrow move into yellow bone marrow, which is then repopulated by pluripotent stem cells. Okay, as you can see in the formation of blood cells, the diagram which is shown over here, the pluripotent stem cell, which is a part of the red bone marrow, it gives rise to two types of or two lineages of the stem cell. First lineage is the myeloid stem cell, and second lineage is the lymphoid stem cell. Okay, so this myeloid stem cell, it can give rise to some progenitor cells. So these green colored cells, they are called as progenitor cell, that is cfue, cfu meg, and cfu gm. And some of the myeloid stem cells, they can directly give rise to the precursor cells. Okay, so you can see uh, the color which is given over here, that is precursor cells. So this cfu stands for the colony forming unit, colony forming unit. Okay, so this cfue, which is a progenitor cell, So what is the speciality of these progenitor cells is that these progenitor cells they lose the capacity to reproduce themselves. Okay, and they are committed to giving rise to more specific elements of the blood. If you can see over here, in this condition, the pluripotent stem cells, as it has capacity to give rise to more differentiated type of cells, like in this case it is giving rise to the myeloid stem cell as well as it is giving rise to the lymphoid stem cell. But when these myeloid stem cells, when they give rise to these progenitor cells, they cannot reproduce themselves. Okay, and also they can only give rise to the more mature cells, that is the red blood cell. In case of this, that is cfue, it can only give rise to the red blood cell, it cannot give rise to any other type of cell. So similar condition with the cfu make. M stands for the megakaryocyte. Okay, so mega means big. Okay, so here cfugm, that is granulocyte. G stands for granulocyte, M stands for monocyte. Okay, so as this progenitor cell gives rise to the granulocyte as well as the monocyte, that's why it is named so, that is granulocyte, monocyte. Okay, so in this we can see this cfue gives rise to the proerythroblast. This proerythroblast is a precursor cell, or when the suffix is blast, it is known as it indicates, or it indicates it is immature cell. Okay, so when suffix becomes site, it is, it means that the cells are mature. But if the suffix is blast, then the it means that the cell is immature or the precursor cell. And the when the suffix becomes site, it means that it is the final cell or it is a developed mature cell. Okay, so you can see in this case the proerythroblast, the suffix is blast, so it is a precursor cell, and this proerythroblast gives rise to the reticulocyte. So you can see the site as the suffix, so it is a mature cell, and this reticulocyte gives rise to the red blood cell, which is alternatively known as erythrocyte. Okay, so this cfu make, it is a progenitor cell for the megakaryoblast. Okay, so which is a type of precursor cell. So this megakaryoblast then gives rise to the megakaryocyte, which is a mature cell, and this megakaryocyte then breaks down into the fragments to give rise to the platelets. Okay, so this cfugm, which is a progenitor cell, then further gives rise to the one, two, three, four different types of precursor cells. So the first precursor cell is eosinophilic myeloblast. Okay, so uh, there is a type of dye or stain that is called as eosin. So eosinophilic means eosin loving myeloblast. Okay, so there is second type, that is basophilic myeloblast. So this basic stain. Okay, so this nucleus of it or the cytoplasm of it, it gets stained with the basic stain, that's why it is called as basic stain loving myeloblast. Okay, then there is simple myeloblast, which is neutral. Okay, this is acidic dye, this is basic dye loving, and this is neutral cell, that's why it is only myeloblast. Okay, and there is another, that is monoblast. Okay, so this eosinophilic myeloblast, it further gives when it develops into the eosinophil. Okay, this basophilic myeloblast, it gives rise to the basophil. This myeloblast gives rise to neutrophil, and this monoblast gives rise to monocyte, which further develops into macrophages. Okay, so here the M can be the macrophage, or it can also be called as monocyte. Okay, so that's all about the myeloid stem cell lineage. So one more thing I forgot. So this myeloid stem cell, it also gives rise to another type of cell, that is called as mast cell. Okay, so this mast cell, it is also known as the histaminocyte. Okay, why it is called as histaminocyte, we will study it later when we will study all of this. Okay, so this is only to let you know that how pluripotent stem cells gives rise to different types of formed elements of the blood. Okay, so as we move forward, we are going to study in detail each and every type of the cell. Okay, so that time we will discuss about the structural peculiarities of each and every cell. Okay, so next, second lineage of this pluripotent stem cell, it is lymphoid stem cell, or it gives rise to the second type of stem cell, that is lymphoid stem cell. So this lymphoid stem cell, it can give rise to the three different types of precursor cells. So the first precursor cell, it is T lymphoblast, second is B lymphoblast, and third one is NK lymphoblast. Okay, all of these three are the uh, precursor cells. So this T lymphoblast gives rise to T lymphocytes, and the NK lymphoblast give rise to natural killer. NK stands for natural killer cell. Okay, but here we have something different, that B lymphoblast give rise to the B lymphocyte, and when the antigen is presented to the B lymphocyte, this B lymphocyte turns into or gets converted into plasma cell, and this plasma cell is responsible for the production of antibodies. Okay, so if we study the immunity chapter, I guess you will have this immunity chapter in microbiology. Okay, there you will study in detail about how the adaptive immunity works. Okay, so there you will have reference to all these type of cells, that is T lymphocytes and B lymphocytes. Okay, so let's see what is erythropoiesis. So erythropoiesis, or formation of erythrocyte, or formation of RBCs, that how RBCs are formed. Okay, so erythropoiesis, it is nothing but production of mature erythrocytes of the peripheral blood, which takes place in bone marrow. So location of production of RBCs is bone marrow from morphologically unrecognizable hematopoietic stem cells. So the pluripotent stem cells which we have just seen in last diagram, they give rise to the these erythrocytes, and it is said so that is morphologically unrecognized. Morphologically means structurally the erythrocyte, they doesn't look any like of that of the hematopoietic stem cell, but still this hematopoietic stem cell is responsible for the production of erythrocytes. Okay, so the red cell production is influenced by growth factors and hormones, notably the erythropoietin. Okay, so this erythropoietin is essential for the process, that is formation of RBCs, and as this erythropoietin stimulates the formation of the red blood cells, that's why the process is known as erythropoiesis. Okay, so erythropoiesis activity in the body is regulated by erythropoietin, which is produced in response to hypoxia. Hypoxia means what? It is lack of oxygen, okay, or deficiency of oxygen. So if uh, a deficiency of oxygen is gets created at any particular in any particular organ of the body, then uh, it will stimulate the erythropoietin release from the kidney. Okay, so kidney is the principal site of the erythropoietin production. So this erythropoietin which is produced from the kidney is responsible for the stimulation of the erythropoiesis, that is formation of erythrocytes. Okay, so this is the genesis of the erythrocyte. So the whatever we have studied in earlier, that is cfue give rise to the proerythroblast, and there on we are going to study how this proerythroblast give rise to the red blood cell. Okay, so before to this there is cfue. Okay, so this cfue gives rise to the proerythroblast. Okay, so the first cell that can be identified belonging to the red blood cell series is the proerythroblast, which is shown at the starting point in the figure. Okay, so under appropriate stimulation, large number of these cells are formed from the cfue stem cells. Okay, so what is important over here, that if stimulated, then only the CFUE will give rise to the proerythroblast. Okay, unless and until there is presence of stimulation, these cells won't be formed. But if there is stimulation, then we will get proerythroblast from CFUE. So once proerythroblast has been formed, it divides multiple times, eventually forming many mature red blood cells. Okay, so whenever it is dividing multiple times, in between we will have these immature cells, that is basophil erythroblast, polychromatophil erythroblast, and orthochromatic erythroblast. Okay, so the first generation cells are called as basophil erythroblast. Why? Because they stain with the basic dyes. Basic dyes means another word for stain. Okay, so with basic stain as they die or they color, that's why they are called as basophil erythroblast. So the cell at this time is accumulated very little hemoglobin. So you should remember, as we move forward you will try to correlate this, that amount of hemoglobin in the cell. Okay, so in the succeeding generation as shown in the figure, the cells become filled with hemoglobin to concentration about 34%. The nucleus condenses to small size and its final remnant is absorbed or extruded from the cell. Extruded means it is removed from the cell. Okay, please mute yourself. Okay, at the same time, the endoplasmic reticulum is also reabsorbed. So why all these organelles are getting removed from the cytoplasm of the cell? The reason is that the they are making way, or they are making space to accumulate the rising amount of hemoglobin in the cytoplasm. If all of these organelles are present in the cytoplasm of this cell, then what will happen? The hemoglobin, the very less amount of hemoglobin can be uh, occupied or can be accumulated in the cytoplasm. So in order to accumulate lot amount of the hemoglobin, all of the organelles they are being removed, even the nucleus is being removed. Okay, so the cell at this stage is called as reticulocyte, because it still contains small amount of basic basophilic material consisting of remnant of the Golgi apparatus, mitochondria, and few other cytoplasmic organelles. So during this reticulocyte stage, the cells pass from the bone marrow. Please understand, when the proerythroblast turns into the reticulocyte, this from this till this sorry till this, that is from cfue to orthochromatic erythroblast, all the steps happen in red bone marrow. But after this orthochromatic erythroblast converts into the reticulocyte, it leaves the red bone marrow and it gets into the systemic circulation. Okay, so the cells pass from the red bone marrow into the blood capillaries by diapedesis. So diapedesis is the process by which the these cells, they squeeze through the pores of the capillary membrane. So whenever we will study the structure of the blood capillaries, that time I will tell you the difference between the blood capillaries and the other blood vessels. Okay, so the remaining basophilic material in the reticulocyte normally disappears within 1 to 2 days, and the cell is then a mature erythrocyte. So once this reticulocyte enters into the systemic circulation and enters into the systemic circulation by means of the diapedesis, within 1 to 2 days, all the cytoplasmic organelles, they get removed from the reticulocyte, and the they get converted into, or they turn up into the erythrocytes. Okay, so next is how the red blood cell production is regulated by erythropoietin. Okay, so in first slide what we have said, that production of erythropoietin is stimulated by hypoxic condition in the organ or in cell. Okay, so let's see that in this it is negative feedback mechanism. Okay, so hematopoietic stem cell we have already studied, that is HSC. It gives rise to the proerythroblast and gives rise to the red blood cells. Okay, and they cause, or they facilitate tissue oxygenation. So in which condition this tissue oxygenation is decreases, that first is low blood volume. If the blood volume decreases, in that case oxygenation also decreases. When there is anemia, we are going to study what is anemia in next few slides, so that time I will discuss. The next is low hemoglobin, that is the amount of hemoglobin present in the red blood cell. If it gets decreased, then it leads to decrease in the tissue oxygenation. If there is poor blood flow to any of the organ, then also tissue oxygenation will be hampered. So this poor blood flow, it may be because of many reasons. So we are, when we will study the pathophysiology as a course, that time we will study about this. Okay, then next is pulmonary disease. Pulmonary disease, when we say, it is related to the lungs. Okay, okay, so pulmonary disease related to lungs. And as we know, the blood is sent from the right ventricle of the heart to the lungs for the oxygenation purpose. Okay, so if there is any pulmonary disease present, that is diseases related to lungs, then the oxygenation of the blood, it gets hampered. That means there is decreased oxygenation of the blood, and as there is decreased oxygenation of blood, the blood which leaves the lungs, it carries less amount of oxygen than it is supposed to be. And as it carries less amount of oxygen, wherever that blood goes to whatever tissues, the tissue oxygenation also decreases, and this decreased tissue oxygenation, it leads to production of erythropoietin by kidneys. And once this erythropoietin is created, it will stimulate hematopoietic stem cells to undergo mitosis, give rise to the proerythroblast, which then finally gives rise to the red blood cells. And the increased number of red blood cells in the body further will enhance the tissue oxygenation, which will give negative feedback to the kidney, so that kidney will stop erythropoietin production, and there will be no further production of the red blood cells. So this is nothing but the negative feedback mechanism by which erythropoietin regulates the red blood cell production. Okay, so let's see about the red blood cells or the erythrocytes. So these are biconcave disc with diameter of 7 to 8 micrometer, okay, or microns. So biconcave shape, you can see concave shape. So this dish shape, so you can see it is a disc shaped structure, and which is biconcave in nature. Bi means on the both side you can see the concave shape. Okay, so in females, the average value of the erythrocyte, or average number of erythrocyte present, it is 4.8 millions, and in males it is 5.4 millions. So let me tell you, 1 million is equal to 10 lakh. Okay, so 4.8 million means multiply it with this 10 lakh, 48 lakh cells in 1 microliter of blood sample. Okay, so see, the number of erythrocytes, it is very high. Okay, that's why they are most abundant cells among the formed elements of the blood. Okay, so the plasma membrane, it is very strong and flexible, and it allows them to deform without rupturing as they squeeze through narrow capillaries. Okay, so the capillary size is very narrow, the diameter is very small, but as they have very strong and flexible plasma membrane, they can easily squeeze through it. Okay, so in the surface or in the plasma membrane, we have glycolipids, and these glycolipids act as antigens, and depending on this presence of antigens, we have our blood grouping system. Okay, whatever blood groups we know, that is O positive, A positive, it is because of these glycolipids in the plasma membrane, which act as antigens. Okay, we are going to study in detail about those, so don't worry about it. Okay, so mature RBCs, they lack nucleus and other organelles. Okay, so they neither reproduce nor carry on extensive metabolic activities. Okay, so there is no reproduction in the mature RBCs. Okay, so the after, as they don't reproduce and they don't carry on extensive metabolic activities, they have definite life period, that is of 120 days. Okay, so after 120 days, they are killed out of the circulation. The next is the cytosol of it. It contains hemoglobin, sorry, they contain hemoglobin molecules. Each RBC contains around 280 million hemoglobin molecules. We are going to study in detail the hemoglobin structure in next few slides. Okay, so the physiology or the function of the RBC, the advantages of it, that is because one second. This some of the structural features which we have mentioned earlier, they provide some physiological advantages as following. That is, as the mature RBCs, they don't have any nucleus. All of the internal space is available for oxygen transfer. Also, they lack mitochondria, so they generate ATP anaerobically. Anaerobically means without oxygen. So as they produce ATP anaerobically, they don't use up the oxygen which they are carrying. Okay. So next structural feature that provides physiological advantage is the biconcave disc shape. So it facilitates its function, that is greater surface area is available for the diffusion of the gas molecules. Okay. So next structural advantage, structural feature that provides physiological advantage is presence of hemoglobin. So it helps in the oxygen transport. So it binds to the iron molecule, and it also transport about 23% of the total carbon dioxide by binding to amino acid, then waste products of the metabolism, and also plays a role in the regulation of blood flow and blood pressure. Okay. So next, let's see about hemoglobin and its formation. So this is the structure of hemoglobin. You can see there are chains in it, and at the center of this you find an iron containing heme particle. Okay. So it consists of protein called as globin. Okay. So you can see, you can divide this into two, that is hemo and globin. So globin is the protein part and heme is the iron containing part. So this globin protein, it consists of two alpha and two beta chains. So this is the usual structure of the globin. So these, please understand this thing, these chains, that is alpha and beta chains, they are made up of amino acids. Okay, amino acids. So a ring like non-protein pigment called heme is bound to each of the four chains. So this heme is a non-protein pigment. Why it is called as pigment? Because it is colored thing. Okay. So this is whatever shown over here, it is heme. Okay. So at the middle of it, it is iron. Okay. At the center of each heme ring is the iron. Okay. So you can see this red color thing is the iron containing, and this ring structure, it is heme, non-protein pigment, and this blue color thing, these are the alpha and beta chains of the globin protein. So next is hemoglobin synthesis. So synthesis of hemoglobin begins in the proerythroblast, which is the first stage in the erythrocyte formation process. So it starts in proerythroblast and it continues even into reticulocyte stage of the red blood cells. Okay. So therefore, when reticulocytes leave red bone marrow and pass into bloodstream, they continue to form minute quantities of hemoglobin for another day or so until they become mature erythrocytes. Okay. So uh, what is the message over here, that once the reticulocyte convert into the mature erythrocyte, they stop preparing hemoglobin. Before to that, the hemoglobin synthesis is still happening in the cytosol, but after they become mature erythrocyte, the hemoglobin synthesis stops. So this is the process we are going to explain. That is, first step is 2 succinyl coenzyme A combine with two glycine molecules to form a pyrrole ring structure. Okay, so such four pyrrole ring structures, they combine together to form protoporphyrin 9. Okay, so in third step, this protoporphyrin 9, it combines with ferrous to give rise to the heme molecule, which is non-protein pigment. This heme combines with the polypeptide chain to give rise to the hemoglobin chain, that is alpha or beta. So such two alpha chains and two beta chains, when they combine together to give rise to the hemoglobin A. Okay. So it is explained in this step only. So succinyl coenzyme A, from what is the source of it? That is Krebs cycle. You have already studied this. It binds with glycine to form a pyrrole ring structure or pyrrole molecule. So in turn, these four pyrrole molecule combine to form protoporphyrin 9. Okay, this protoporphyrin 9 combines with iron to form heme molecule. So each heme molecule combines with a long polypeptide chain. Polypeptide means what? The amino acids. The amino acids, they are joined together with a peptide bond. The bond which is present between the two amino acids, it is called as peptide bond. And as there are many of such bonds in the amino acids that form this chain, that's why it is called as polypeptide chain. So a globin synthesized by the ribosome. So what is the site where the globin is synthesized? It is a ribosome. So it forms a subunit of hemoglobin called hemoglobin chain. Okay, so by this process we get alpha chain and the beta chain, and when such two alpha and plus two beta chain are combined, it gives rise to hemoglobin A. Okay. So next, depending on the amino acid composition of the polypeptide portion, there are various types of hemoglobin chains. So these chains are designated as alpha, beta, gamma and delta. Okay, so apart from the alpha and beta chain which we have studied earlier, there are gamma chains as well as delta chains, and the amino acid composition of this gamma chain and the delta chain, it is uh, different from that of the alpha and beta chains. So the most common type of hemoglobin is hemoglobin A. So it is most common form in a adult human being, and it is combination of two alpha chains and the two beta chains. Okay. So we will study about anemia in next lecture. So let's finish it over here only. Okay, thank you.